Hello everyone, I am based at University of Tasmania in Australia and I've been here for the last 20 plus years and it's been a really good experience working through different scientists and colleagues throughout the world. And I would like to share with you today, actually Ramesh asked me whether I could present something on marine biotechnology to you guys on the occasion of this biotechnology Vignan skill workshop that he has organized. Initially, I was a bit intimidated because marine biotechnology is as vast as the ocean. As you know, oceans are very vast. Because it is a bit out of my research expertise, my research expertise is more on marine pests and fish diseases. So I have decided, in order to give you some background about marine biotechnology, not that I don't understand it, but it's just that it's a bit removed from my research area. But however, I have taken the time to give you some background, or I'll take the time to give you some background on marine biotechnology and present a bit more of my own research interests so that it actually gives you some background on what happens in the big broad sphere of marine environment. Okay, with that note, I would start. Come on. Okay, it's up here. Okay. Does anybody recognize this organism? Participants, can they speak or not? Can they speak? Can the participants speak?
They can.
We have muted them, sir.
Box.
Okay. I mean, if you can, if you know what this organism is, you can either write it in the chat box. Never mind. I'll tell you. This is a marine microscopic organism called Dinophysis. It is a planktonic organism. Looking at its morphology, Alistair Hardy, a famous marine biologist, is supposed to have said, man is unwittingly copying the inventions of nature, because he had already seen how helicopters work, and post that he had seen this particular organism, and then he made this comment. Had he seen this organism before, he would have said, man is copying the inventions of nature, not unwittingly copying the inventions of nature, because this particular organisms, as you see on the top there, can you see my cursor? Can you guys see my cursor? No.
No, sir. Sir, no. It's not visible, sir.
Let me see if I can activate the cursor. Never mind. There are two panels. One is at the bottom right is a panel. The panel is of an electron microscope picture. On the top is a light microscope picture. On top, what you see is a world like similar to helicopter. It's pretty much a propeller-like apparatus that helps these organisms to go up and down the water column. So it is amazing.
Regarding that's good, sir. Sir, somebody written dinoflagellates.
Yes, it is a dinoflagellate, but it is specifically Dinophysis. Very good. Dinoflagellate is a very good answer. And it goes up and down to the water column with the help of this propulsion. So it's pretty much similar to what you see in a helicopter. So marine organisms have a lot of technology to offer to mankind. Just to remind, not that you guys don't know, just to remind that we are a blue planet, because 71% of the water, 70% of the Earth is covered by water, and land mass is relatively tiny. In the context of biotechnology, here is a definition of biotechnology by FAO and OECD, which is an all-encompassing definition. Essentially, it says application of science and technology to organisms from marine resources, as well as parts, products, and models thereof. So it pretty much includes everything, anything that you take from marine organisms and put it into different organisms or into marine organisms themselves to produce knowledge, goods, or services. It is a marine biotechnology. So it's a very broad and encompassing definition. Does anybody recognize this picture? Yes? Doesn't matter. Again, it's what we know. It's what we, the subcontinent people, know as Samudra Mantana, which is essentially churning of the ocean by suras and asuras to produce the elixir of life, the Amrit. Right? So this has got a significant connection to marine biotechnology because it is churning the ocean to produce the elixir of life. So it's mythologically quite linked. Marine biotechnology is mythologically quite linked to Indian culture. The key lessons from this is that of inclusion. As you know, in order to churn the ocean, it required both suras and asuras to collaborate. So collaboration, inclusion, and sustainability are significant lessons from this picture. And there's also an element of caution, simply because, as you know, that when they churn the ocean, they not only produce amrita, they also produced what's called halala or nila, or the poison, which Shiva is supposed to have drunk. So there's always a problem associated with any solution. So that is a caution that we must be aware of whenever we are developing any technology. Okay, with that note, going on a more positive note, ocean, as you know, or ocean or seas, as you know, are a vertical production system. They not only offer the surface, but they also offer a great depth, as you see from this picture where mussel rafts are hung from the surface, where mussels can grow all through the column. So that means it gives great depth and great potential for growing anything in the ocean, unlike on land. On land, at best, it's a monolayer. Here are some good examples of well-established examples of outcomes of marine biotechnology from the oceans. One of the first one is the enzyme called Taq polymerase, which I'm sure you all know what is PCR. PCR uses an enzyme called Taq polymerase, which is actually isolated from hydrothermal vents from microbes from the oceans because it is heat resistant. It's active at 72 degrees, therefore it can tolerate heating and cooling, which is part of a PCR reaction. And the second one is a green fluorescent protein isolated from a jellyfish. And again, it's an integral biomarker in most of the biotechnology applications in generating transgenic animals, which acts as a very useful reporter on a number of aspects. On aspects such as genetic transformation, on aspects such as changes in gene expression and the like. So it's an integral part. Both the Taq enzyme and the green fluorescent protein are what I call a key workhorses of modern biotechnology. So they both come from the marine environment. Similarly, there are a number of other products, and far before that, we know that there was a compound called C factor, which is isolated from horseshoe crab, which is an integral to detection of endotoxins in blood and food products. So there are a number of products which have come from marine environment that are pretty much integral to the societal usage on a day-to-day basis. What is less utilized from the marine resource and which is vast in quantity is the marine algae. However, this is now slowly changing. Over the last 10 years, the marine algae have become an important source of resources that are attracting a great deal of attention from both researchers as well as industries. And this is particularly relevant for India, simply because India is predominantly a vegetarian society, despite that we have very little history of using marine algae as human food. Right. So, however, hopefully in the next few slides, I'll try and convince you that there are significant resources from the marine environment that could be used both by vegetarians and community at large. Okay, here are the examples of products that have been developed from microalgae more recently. It's a big source of bioethanol. It's a supplement for fuel, a fuel supplement for essentially a fuel supplement. And what is more important is bio oils. As you'll be aware, from the marine sphere, we'll be more familiar with using cod liver oil pills and the like. But however, most fish do not produce these essential fatty acids. It's actually the small microbes that actually produce these essential oils that are bioaccumulated in fish, from which we are used to extracting or utilizing the resources generally. But it is the microorganisms, the microalgae, that are the primary source of production of these polyunsaturated fatty acids, which are essential for all living beings. The other parts are proteins. They're also significant source of proteins, which now have found way into the form of pills, both as supplements for animal feed as well as human human consumption. The more attractive part of bioactive compounds or extracts from marine algae are the pigments, such as beta carotenes and the like, which are also antioxidants that are increasingly being extracted from marine algae and put into feeds, and also as directly developed for human consumption. Here's a slide showing some of the work done by my colleague Kim Lee Chang at Australian National Collection for algae. As you'll see from the picture, the breadth of color is amazing. It encompasses the whole spectrum of rainbow. So as you see from this panel, you can see all the different colors. So this can be, the way these are actually propagated are, first they're isolated into individual cultures, and then they're propagated into large-scale production systems. As you see on the far right, they are actually produced in large barrels. So these production systems can be done on land or at sea. Here are large-scale production systems in Australia where microalgae are produced in industrial scale. One at Myrtles Raceway Pond in Wyoland, South Australia, and the other one is a production of Dunaliella for production of beta carotene for incorporation into animal feed and for human consumption. So these are significant pharmaceuticals that are now already found into the market, found their way into the market. Okay. We are, however, less prone or less prepared to farm the oceans as yet, because the technology is not yet there, but we are far more familiar in farming the land. So the question arises, can we take systems that are in the sea and can we put them into land-based systems? The best example is a transfer of PUFA pathways into, for example, in canola. Canola, for example, doesn't produce polyunsaturated fatty acid. There are some plants such as flax that produce PUFAs, omega fatty acids, but they produce in small quantities and they're less efficient in production, whereas the marine microalgae are highly efficient in producing and synthesizing PUFAs. So understanding the biochemical pathways in the marine algae and then transferring those biochemical pathways into land plants could enable us to produce the PUFAs more efficiently on land. So that is the example here, trying to take the pathways in the marine systems and transferring them into land plants. So this is not just a hypothesis, it's actually happening as we speak. As you see in this panel, there are a number of enzymes that are involved in the production of PUFAs, that is DHAs and DPAs, have been already isolated and have been transplanted or have been transferred into canola plants. Now we now have canola crops that actually produce PFAS at higher levels, and that is all relies on the enzymatic pathways that have been transplanted into canola. Not only that, now these have already found way into the market. Okay, one of the important bits, I suppose, is, as I said, India is a nation full of vegetarians, so we are generally less keen to accept PFAS from fish source, for example. So you don't have to eat the PFAS from fish, but if you were to actually produce them in canola, you'd be able to. They are a direct source of vegetarian source of PFAS that are pretty much essential for development. I'll probably stop there for a while. If there are any questions, I'm happy to take at this point of time.
ಏನಾದರೂ ಕ್ವೆಶನ್ಸ್ ಇದ್ರೆ ಕೇಳಬಹುದಲ್ವಾ ಪಾರ್ಟಿಸಿಪೆಂಟ್ಸ್? You have any questions till now, can write on chat box.
Is somebody looking at managing the chat box?
Yeah.
Is there anything, Srinivasa?
Sir, no, sir, no questions so far, sir.
So far?
Yes,
You want
sir.
to give them a minute if they want to ask anything?
Yeah, that's what I am writing them. So they will ask in the end, sir.
Okay. Okay, if that's the case, then we can hold them till the end. So I'll now move on from microalgae to macroalgae. Okay. Here are pictures of 6 prominently edible seaweeds. There are not so much, because again, I said we don't have a history in India, for example, of
Sir,
eating
one
these
question,
seaweeds,
sir.
right?
Oh, sir, one question.
Sure.
They asked that what is oil, TAG?
ITAG,
Yes, sir.
in the context of this slide?
I think so, sir. Yes, sir. Is it, and one more question, sir.
Yeah.
No, there is little bit of confusion. Is the question is not clear.
Okay.
Let him ask again.
Sir, one more question.
Sure.
Is it possible to degrade plastic by using marine algae or fungi?
Not so much marine algae, but there are a lot of marine microorganisms that can be used to degrade plastics. And good question, very good question. This algae are a good substitute for plastics. There is increasing research of where people are trying to use marine microalgae to produce alternatives to plastics. Right?
Yes, sir. Yes, sir. So you can carry on now, sir.
Okay. What was the other question? IT?
Sir, oil, just they put bracket. What is oil? They put bracket and TAG tag.
Okay. I think. Is this the one?
Yes, sir.
TAG, yes.
Yeah.
That's pretty much for different applications.
Yes.
Okay, for agriculture and the like. So there are different applications. Okay. Let's move on to edible seaweeds. As I was saying, seaweeds are very popularly consumed in parts of Japan, China, and the Far East, but not so much in India. I can't recall we having any historical preference of eating seaweeds. I'm happy to learn if anybody knows about it, but otherwise, there is significant potential to use seaweeds. Again, They are also primary, so they can produce PUFAs, and they are a very good source of iodine, for example, and they've got lots of health benefits. And that is pretty much evident in the longevity of longevity and non-obesity of people in cultures such as Japan. Okay, they can not only be eaten directly, but again, they are a great source of discovery or mining for bioactive compounds. Again, this slide summarizes the several different bioactive compounds that have been already extracted from seaweeds that have been now part of food industry, cosmetics, as well as pharmaceuticals. And the most recent development is a species of marine algae called Asparagopsis. It's a red algae, and it's been proven that it has an ability to reduce the production of methane in cows. If it is so, feeding Asparagopsis by itself is not an option because it is not because cows can't digest it very efficiently. However, substituting it in small quantities is proven to reduce the production of methane by cows. So it is expected to directly help in combating the effects of global warming. So this is an emerging science, and the compounds that are responsible, compounds as well as the microbes that are responsible for reduction of the methane are now a significant discipline of study in marine biotechnology. Just to give a commercial background, if you like, commercial context, the use of marine products can be divided into three different groups: those that can be used immediately, those that can be used after certain development, and those that require a long-term development. As you see, it's quite obvious the bulk chemicals, the bulk enzymes, animal feed, etc. are direct extract resources. They can be used immediately. There's very little investment required. There's very little time required, and hence, however, the downside of that is they offer very low economic profitability. However, if you invest significant resources into research, you can develop high-end products such as cosmetics, nutraceuticals, enzymes, and fine chemicals that takes somewhere between 8 to 10 years to develop before they reach market. And then at the very high end, you have the pharmaceutical and medical applications, which requires not only development, but also they require significant clinical assessment before they can become an accepted product. So they take much longer time. However, the risks are higher there, as well as the rewards are also higher. And this applies to any biotechnology product. Okay, so that pretty much ends my introduction to general marine biotechnology. Before I head into the problem of aquatic pests in marine environment, again, I'm happy to take questions on the aspects I presented so far. Srinivas, any questions?
No, sir. So no questions.
Okay.
Please.
Just give a minute, and then we'll go on to aquatic pests.
Yes, sir. Please.
Nothing in the chat box yet?
Nothing, sir.
No?
Yes, sir.
Please feel free to tell me if it is too cryptic to understand. If so, then I'll try and make it less complex. Because particularly when we are in research, we tend to think that everybody knows everything else, right? So So if you think anything is complex or needs to be toned down, feel free to tell me. You're good, Srinivas? No more questions?
No more questions, please, sir.
Okay, we'll go to the aquatic pests. As I was saying, this is primarily my research focus or interest, if you like. I've been involved in research on marine pests for nearly 20 years, right from developing genetic solutions to physical management and eradication of pests from large ecosystems. As you see in this panel, there are a number of pests that I've presented. One is an Asterias, which is sea star, which is a pest in Australia. There's a fanworm, Cebalid fanworm, again a pest which fouls hulls of ships and the like. And on the far right is an European crab, again a pest in Australia.
Sir, one question is there, sir.
Yes. Go on.
Yes, sir. It's uses of marine algae, but how about other algae which are dangerous for human too?
Sure.
From Devraj K. Yes.
Yeah. Again, good question. Like any compound, any land-based or marine-based products, some of them are harmful. There are a lot of harmful algae which can produce paralytic shellfish poisoning. So we need to be aware of that. So that's an essential component of seafood awareness of being what is poisonous and what is not poisonous. There are a large number of animals and plants in the marine system that produce toxins. So you need to be careful of them. For example, dinoflies, some of the dinoflies produce toxins, and those get bioaccumulated, for example, in shellfish. And people, when they consume them, they suffer what's called paralytic shellfish poisoning. And another example is a tetrodotoxin produced in certain pufferfish, again produced by microorganisms in the gut microflora of these animals. And they bioaccumulate tetrodotoxin. Again, it's a neurotoxin. However, it is a fine delicacy in parts of Japan where specialized chefs prepare the meat in such a way that it only gives an adequate amount of tipsiness, if you like, and people really enjoy it. It's something like a drug. People use it, although it is a poison, people use it in moderation as a drug. So it's like anything else. If it's in excess, anything can be poison. So yes, marine environment is full of organisms that do produce toxins, and you need to be aware of those.
Yes, sir.
Yeah?
Yeah, please, sir. Yes, sir.
Good. Okay. As I was saying, the significant problem from pests in the marine environment comes from shipping activity. As many might know, ships, when they go from one place to another, they carry cargo. But sometimes there's no cargo. When there's no cargo, the ship needs to balance itself or ballast itself. If there's no weight on the ship, the ship can topple. In order to avoid that, ships take water from the environment. When they take water from the environment, they take large number of microscopic organisms into their hull. And they take these organisms and transport them to different environments where they're completely foreign or exotic. And when they take new cargo, they tend to expel that water. When they expel that water, they release a large number of these microscopic organisms they carry in their belly. So these organisms can go and become established in the new environment and can become pests. So there are over 1,593 such invasive species in the world, in the marine environment, in different environments, in different places. So it's a very big problem created by the shipping industry. And there's very little that many can do to mitigate this other than to try and come up with solutions. So I have been involved in this research for quite some time. The things that we can do is to really detect them early. If you can detect if a particular port or a particular ship is infected with these pests, then we can use what is called mid-sea exchange of ballast water, because when you mid-oceans are less productive, so they have very few organisms. So if you change water in the mid-sea, it dilutes the risks posed by such microorganisms. So in order to, again, changing water mid-sea is a very risky activity and also expensive activity. So you need to do that only if it is necessary to do the exchange. In order to know when to exchange, when not to exchange, you need to be able to predict if a particular ship or a particular port is infected by that pest or not. So if you are able to predict that, then we can manage that very well. So through my work, I was involved with UNDP, which actually oversees the shipping activities, which governs the shipping activities on behalf of the United Nations across the world. And UN came up with a convention in 2004. Essentially, all maritime states are a signatory to this. That means they are bound by this convention to manage the shipping activity. So as part of this, I acted as a technical expert in order to try and come up with methods to detect these microbes using genetic techniques, essentially using gene probes to detect these pests in the ports, in the hulls of the ship, and anywhere else there could be a problem. So that details of that all work is captured in that particular report that we prepared for UNDP. The concept here is if you're able to detect using genetic probes on a platform of microarray, this information can be relayed to the ships on land so that the ship captains can take action as quickly as possible to exchange the water and minimize the risk to the ports they visit. So it's now an integral part of managing shipping activities globally as part of a program called GloBallast. GloBallast is a program overseen by UNDP, which requires that ships and ports monitor themselves for the presence of these microorganisms or planktonic organisms, which could cause a problem in foreign ports. And there are incidences where maritime states have denied entry to ships on detection of pests. So it can be a very expensive exercise for shipping activity if these are not monitored on a regular basis. And biotechnology has got a significant role to play in terms of developing rapid detection kits, something similar to what now you are more used to in terms of the rapid kits for corona and the like. So it's both antibody-based kits and a DNA PCR-based kits. So it is a very, very big discipline. That is for prevention, but I'm also interested in developing solutions for pests, for pests which are already a problem. So for these, the current methods of physical control and chemical controls are not particularly effective because physically it's impossible to reach all the nooks and corners where these pests hide. Unlike on a terrestrial system, in an aquatic system, all these organisms are pretty much unseen, right? So you can't, in an aquatic system, most of the danger is beneath the water, so you can't see them, unlike in terrestrial. So it adds an added difficulty in a terrestrial system. So the best way to reach them and control them is using genetic approaches, which my group has been doing for quite some time. So I'm giving an example of two of the pest species that are significant problem to Australia that we've been targeting. One is called Gambusia, which is on the left-hand side. It's a small, tiny fish. It's actually interesting, a livebearer. It actually keeps the babies just like the humans. It incubates the babies inside. Unlike most fish actually lay eggs, whereas these keeps the baby inside and then gestate the babies inside the mother. And the fully grown babies are delivered just like the humans or other mammals. And on the right hand is the carp. Again, carp is an important source of food in India and many parts of the world. However, it's also deemed as pest in some parts, including Australia. As you see from this graph, it's a problem throughout most parts of Australia. I'm not going to, just was telling you that on the left-hand side, as you'll see, is a male and a female gambusia. Male has a modified anal fin, which it uses to deposit sperm into the female. So it's almost acts as an organ that helps it to deposit sperm, and it somewhat mimics the copulation that happens in mammals. And on the right-hand side, as I say, it's a carp, which is a long-lived species. This might be of interest to you. There was a carp named, a koi carp named Hanako, which supposed to have lived from 1751 to 1977, over 200 years. It had a longevity over 200 years. So this is a very long-lived fish. It's very difficult to get rid of fish that actually got that long-lived. The concept of genetic control has been around for quite some time. It's actually Hamilton who first proposed a concept called spread of Y. So he said, if there's a means of spreading Y preferentially in organisms that have XXXY, I hope you do understand what is XXXY. It is the sex determining mechanism as you see it in human beings. Whereas there are certain animals like birds, chicken, where you've got a different sex determining mechanism, which is called ZZWZ, where female is heterogametic. Whereas in case of XXXY, it's a male, which is heterogametic. X and Y, they're different. X is different and Y is different. Whereas females is homogametic because it has both X and X chromosomes. So that's why we call them homogamous. But X and Y is a different gamete, and X is a different gamete. So they are heterogamous. So the terminology heterogamous and homogamous is very important to know in developing genetic control options. How with the recent advent of molecular techniques, now it's far more easy to develop these techniques. This particular concept had almost remained a theory for a long time. Now that we can actually splice genes, modify genes, we can develop GMO solutions. So it is now more possible to develop these techniques. I have summarized here the different approaches. This can be broadly classified as chromosome set manipulations, which are essentially non-GMO approaches, and others as recombinant approaches, which are classified as GMO approaches. I say this is quite important because GMO approaches attract lots of public scrutiny. They're not easily accepted by public as compared to non-GM approach. So non-GM approaches are typically well accepted. They've been used in the past very much at industrial scale. So there is less issues with non-GMO approaches, whereas the GMO approaches attract certain concern from the society simply because of perceived or real risks. Okay, so taking into all this into account, and again, that I prepared for Inland Fisheries Service, which asked me to do this, which commissioned me to do this in 2012 to try and identify what are the best methods. And essentially, we came up with two methods, which are highlighted in green. One is called the Trojan approach, which is a non-GMO approach. The idea here is if you use these methods, you can drive the population to have one or the other sex. That means when you introduce such animals in the population, they go and breed in the wild and produce offsprings of only one sex. So over time, the population will be devoid of one or the other sex, and hence the population collapses. They can't progress or they can't propagate. Just to recap, I'm sure a lot of you are aware, with the GMO technologies, there are 4 different approaches how you can manipulate. One is called knock-in. It's an example of putting and inserting a new gene into the genome of the host. So that is knock-in, basically knocking the gene into the genome. And the second approach is knockout. Here, you're actually taking an important gene that might be responsible for making an animal a boy or a girl. If you take that gene out, then it might make that animal either a sterile animal, or by default, it could make an opposite sex animal. So you can take a gene out and make it dysfunctional so that it can change a trait. The third option is called knockdown. It's a technology where you knock the function of the gene at a transcription level. That means the gene itself is intact, but the RNA produced by the gene is broken down into pieces using a technology called RNA interference. Again, if anybody's interested, I'm happy to talk about it, but again, it's suffice to know that there are 4 different ways how you can manipulate genes to produce GMO organisms. And the more recent development is what the editing, where you can use very fine tools such as CRISPR or Talon, where you can alter the gene very minutely and yet change their function. Right? Either you can make them dysfunctional or make them to function in an opposite way, or you can even make them to function in a rapid way. So that's by doing editing. I was telling you that there is little acceptance for GMO approaches. So social license is very important, particularly when you're trying to implement such large-scale engineering of ecosystems. So when we did a nationwide survey, it was quite clear. Don't worry about the graphs there, what the graphs are telling, but in summary, there was less public acceptance for GMO, which you would somewhat predict. But this gives a solid evidence that in society, particularly in Australia, there is still a resistance to use GM approaches. So non-GMO approaches are still the most preferred. Given this, we honed in on a non-GMO approach, which is called Trojan chromosome approach. For those who may not know, in fish, we can change the sex of the animals if they are treated with either estrogen or testosterone early in their life. That's immediately after they're born. If you actually treat the organisms with estrogen, even though their genetic sex is male, you can convert them into female. As you see in the panel on the left in the schematic, so as an XY male, all the square boxes represent male and round circles represent females, okay, in that schematic. So if you were to treat estrogen to boys early in life, you can actually convert them into girls. And when these girls with a male genotype, so essentially genetically they're males, but only functionally they're females. So when they mate with boys, say they produce what are called super males or YY males. And these YY males can be eventually made to produce what are called YY females. So the whole idea that when the YY females go into the population, this female case YY, when it mates with an XY, it produces 100% boys. There are no girls produced. So as a result, over time, only boys are produced. So the population is completely devoid of girls, and eventually the population collapses. Okay, so this can be modeled. So here is an example of how the dynamics works over time. So in red is actually the population of those YY females. So if you stock them at a steady level, in pink is actually the population of the girls. The population of the girls the number of girls in the population reduces over time, and there comes a time when there are no more girls in the population. So you can predict and say how long it might take in order to drive a population to extinction. I'm not going to go into all the other details. I'll just skip this. Again, if anybody's interested, you can write to me. Ramesh has my email address. I'm happy to elaborate. As I was talking to you, we have demonstrated that we can do sex reversal in Gambusia, and we can produce wild females in Gambusia. And now we are at the stage of trying to test these at a population level. Okay, sorry, I'll see if I can. Can you see the video?
Yes,
Has it come up now? So just to show you a video, see that these are Livebearers. They actually produce live embryos.
So sorry to interrupt. Could you make it full screen, sir? That video.
Okay. Sorry, I'm not able to do it for some reason. Sorry.
That's fine, sir. Fine, sir.
Hang on. Sorry about that. Somehow I'm not able to maximize that.
That's fine, sir. Fine, sir. Replay it, sir. You can see it.
The point I was trying to make here is that they actually produce babies, not lay eggs, and they show all the traits. They have normal birth, breech birth, and twin births. So you can actually see those. So that's a normal birth with tail first. Can you see that? I hope you can see it. Sorry, I can't maximize the screen. So that's a normal birth.
Jawahar, sir, sorry for disturbing you. If
Sure.
possible, could you put that YouTube link in the chat box so that people can open and watch it in
Okay.
the system? Copy the link and put it in the chat box, sir. Copy video URL. Yeah.
Where's my chat box on? I'm trying to see where my chat box has gone.
It's there on the side. I'll just send you a message, sir. You will get it. I sent you a text in the chat box.
Good.
Yeah, hope you got it.
Yeah. Okay. The link should be there now.
Thank you. Yeah, yes, sir. Thank you.
Good. Okay. I'll probably go to the next one. Have I lost you? Srinivas, have I lost you?
Please continue.
Have I lost you or not? No?
Please carry on.
Okay. Okay. Again, I probably will skip this. The only point I wanted to make is that in case of fish, unlike in human beings, you can actually see the difference between an X and Y chromosome based on the size. We know that X chromosome is bigger than a Y chromosome in humans. So when you do a chromosome spread, you can distinguish them. Whereas in fish, most fish, the sex chromosomes are not different. They're homomorphic. That means there's no difference between the size. So it's very difficult to distinguish them by carrier morphology. Can you hear me? Can you hear me?
Sir.
Okay. So one of the way is to really do what is called progeny testing. So if at all we have successfully sex reversed, that is in a normal mating, and a female with an XX chromosome mates with a male with an XY chromosome, it produces 50-50, as you know. But however, we have a sex reversed female which has an XY genotype. So when you make an XY with an XY, we produce 1 to 2 to 1. So as a result, the moment we get this ratio of 1 to 2 to 1, then essentially what you see in physically is 3 males to 1 female. So once you get the sex ratio, then you know the sex determinant mechanism in that particular fish is XXXY. Similarly, in case of female heterogamety, if a female carries a ZZ and a male carries a ZZ, you end up producing all male population. So it's an easy approach or a foolproof approach to determine the gamete, because determining gamete in fish is far more difficult compared to human beings or other mammals where the sex chromosomes are different in size. What has turned out from our research is that actually Gambusia, I'm sure you're all aware that zebrafish has become one of the key model species for biomedical research. However, our work is showing simply because, like humans, zebrafish gestate the young ones within, and their developmental pattern is also quite similar to humans relative to zebrafish. And so we are now showing that Gambusia might be a far better medical model to study conditions in humans as well as in other animal models. The advantage is that fish produce large number of progeny, so you can experiment far more easily. Okay, for example, we have shown that heart rate of Gambusia is similar to that of males, where there's a difference between sexes in the heart rates, whereas zebrafish doesn't show that difference, sex-specific difference in heart rates. So there are lots of similarities of Gambusia that are far more easily transferable or applicable to human situations. Okay, again, I probably will skip this. Using genetic markers to identify which one are sex reversed. Again, a lot of it is published. In the interest of time, I'll try and go through this rapidly. The other fish I was talking to you about, carp. Again, we developed several different biotechnologies here. Here's what we call the Judas carp, where you implant radio transmitter into the cavity of the fish, and then the fish can be tracked, and then that allows you to capture wild carp. And then we've developed surgical techniques to sterilize fish, something that you don't think it is possible for fish because they're small animals. Indeed, it is possible to sterilize fish using surgical techniques. Sexing fish again using ultrasound. All medical applications hold good for fish, is increasingly becoming very popular. What are we planning to do in the future? So there are biotechnologies where you can, I'm sure you're very well aware that you can store sperm and egg in human situation. In human situation, for example, the eggs do not have yolk, whereas in case of fish have a lot of yolk. Because of the presence of yolk, it's very difficult to store them in refrigerated condition. So alternatively, you can preserve the germ cells, because most of the gonads have what is called germ cells, from which the sperm, for example, sperms and spermatids are produced from the germ cells, and oocytes and ovaries are produced also from germ cells. So you can actually mark these germ cells, as you see in this panel, with the green fluorescent proteins. The germ cells are marked with green fluorescent protein, and then you can actually store the whole testis or ovary in liquid nitrogen, and you can harvest these cells which are marked into then to transplant them to hosts. So these are emerging technologies. I'll probably end with that one in the interest of time.
Sir, sir, there is a question from Basuraj K.
Yeah.
Human activities are increasing marine ecosystems these days. So how can we control or eliminate?
Eliminate the human activities.
Yes, sir.
That's a very difficult question. Probably a question for the Home Ministry. Yes, again, education is an important part, because you are all teachers. So we started very young. So as a teachers, you've got a big role to play to educate young people the principles of precautionary principles of being aware of environmental consequences, just like impacts of marine pollutants. For example, the microplastics are becoming a significant problem in the environment these days. Not so much the large microplastic, but the small microplastic that get used in cosmetics and the like. They easily get bioaccumulated in animals. They not only cause problems in the animals, but they also, when we consume, we actually suffer those consequences. So although we might be thinking that we are not affected, we are somehow affected indirectly. It's like an essential activity that we need to actively control, regulate, but more importantly, educate the young minds. And that is more in your hands as a key science teachers, that you would be able to contribute in a much great way.